Welcome. Today we will talk about enzymatic function. And at the end of today's lecture, we hope to understand the function of enzyme as catalyst, understand the metabolic pathway, the lock and key model, as well as the induced fit model, understand the effects of pH, temperature, and inhibitors on the catalytic activity of enzymes, and go through common examples of enzymes. Enzymes are basically defined as large biomolecules made up of proteins that are used to speed up the rate of chemical reactions. These chemical reactions can either be anabolic or catabolic. Enzymes catalyze specific reaction, hence they need to have the right shape in order to bind with the substrate. In this figure, the enzyme has this shape with which the substrate is able to bind onto. Enzymes are usually named for the specific reaction. Examples include sucrase enzymes breaking down sucrose, proteases breaking down proteins. So you can identify the function of the enzyme from the name of the enzyme. It is important to note that the activity of enzyme is affected by pH, temperature, as well as the presence of inhibitors and the concentrations of substrates. There are two major types of metabolic pathway. We have the catabolic pathway, which refers to the breakdown of complex molecules into smaller or simpler molecules, and it is usually followed by the release of energy. Anabolic reactions, which is the opposite of catabolic reactions, is the build-up of smaller molecules or simpler molecules into larger or complex molecules, and this demands energy. A typical example of a catabolic reaction is the process of digestion. Example of an anabolic reaction is the growth and bone mineralization within the cells of the human system. Energy is needed for any chemical reaction, and the energy needed to initiate the chemical reaction is referred to as the activation energy. The function of enzyme as catalyst is to lower the activation energy and speed up the rate of the metabolic reaction. However, the enzyme itself is not consumed in the chemical reaction. Before we go through the models underlying enzymatic function, let's define some terminologies. Substrate. It is defined as the substance or the molecule that the enzyme interacts with. Products are the new molecule formed out of the interaction between the enzyme and the substrate. The active site is the region or the part of the enzyme that the substrate binds to for catalysis. One of the models underlying enzyme reaction with substrate is the lock and key model. The lock is the enzyme and the key is the substrate. Only the correctly sized key will be able to fit into the active site of the enzyme. The specific shape of the enzyme and the shape of the substrate is what enables the substrate and the enzyme to bind together. And this is a perfect fit between the substrate and the active site. Another model is the induced fit model, and it is based on the flexibility of the enzyme structure. Here, the active site of the enzyme continually reshapes till it fits into the structure of the substrate. Temperature affects the activity of enzymes. Even though rising temperatures speed up the rate at which the enzyme functions, beyond the optimal temperature, the enzyme begins to denature, and this declines the activity of the enzyme, and finally the enzyme stops working. Another factor that affects the enzyme function is pH. Every enzyme has its optimum pH value, and beyond this pH value, the enzyme shape denatures and the activity of the enzyme declines or ceases. The function of buffers built into organisms is to prevent enzymes from exceeding their optimal pH levels. Another factor that affects enzyme activity is inhibitors, and they alter the catalytic action of the enzyme. The inhibitors bind to or block the active sites of the enzyme and prevent the enzyme from interacting with the substrate. Some inhibitors are reversible, while others are not. Common examples of enzymes include lipids, found in meat and dairy products. It breaks down fats into glycerol and fatty acids. Another enzyme is the catalase enzyme, which can also be found in potatoes. It breaks down the hydrogen peroxide into water and oxygen. The last enzyme for this lecture is the lactase, which can be found in milk. It breaks down lactose sugar into glucose and galactose. The takeaway for this lecture is that enzymes serve as catalysts by lowering the activation energy in order to speed up the chemical reaction. The two main models underlying enzymatic function is the lock and key model and the induced feed model. It is important to note that enzyme activity can be affected by pH, temperature, and the presence of inhibitors. I hope you enjoyed this lecture. See you next time.